thing we'll discuss in this lecture is how a blood supply can be delivered to the medulla without washing out the medullary interstitial concentration gradient. There has to be a blood supply, of course, in order to deliver oxygen and nutrients and remove waste and excess solutes and water that have been reabsorbed from the tubules. But this has to be accomplished without flushing out the medullary interstitial concentration gradient. We will then take a walk through the tubule based on everything that we've discussed so far and see how in the presence of antidiuretic hormone, a concentrated urine is generated. We'll also look at how a dilute urine is formed in the absence of ADH. And it's important to note that in both cases, the fluid arriving in the beginning of the collecting duct is going to be dilute relative to plasma. We'll then speak a little bit about the molecular mechanisms by which antidiuretic hormone controls water permeability in the collecting duct. Just a reminder, the medullary interstitial concentration gradient is created by two processes. One, active sodium chloride reabsorption from the thick ascending limb of the loop of Henle. That's the NKCC2 transporter. Some of the sodium chloride gets trapped in the interstitium, and this involves what's referred to as countercurrent multiplication of a single event. The single event is reabsorption of sodium chloride, and the countercurrent multiplication involves the increased concentration of sodium in the fluid delivered to the thick ascending limb due to events in the descending limb of the loop of Henle. This process is always present to some extent. The second process is passive urea diffusion and recycling. And this only occurs when the antidiuretic hormone is present because that's the only time that the inner medullary collecting duct is permeable to urea and allows outward diffusion or reabsorption, some of which gets trapped in the interstitium and some gets recycled into the thin ascending limb of the loop of Henle. So again, we have multiplication of the single event. In this case, it's passive urea reabsorption. What we're going to talk about now is how the medullary interstitial concentration gradient is maintained. And this is possible, one, because vasorectal blood supply to medulla is slow, and that's a very important point. But the most significant is the anatomy of the vasorectal blood vessels, because these also form a countercurrent. But here, there's countercurrent exchange. It's an important distinction between the countercurrent multiplication that's going on in the tubule, which is creating the medullary interstitial concentration gradient, as opposed to the countercurrent exchanger, which is occurring in the vasorector, and you will see that that permits maintenance of the gradient while at the same time allowing perfusion of the medulla. First of all, let's just recap on some of the characteristics of renal blood flow. Renal blood flow is extremely high. The kidneys are very small organs, and yet they're receiving about 25% of the cardiac output. Of that high blood flow, most is going through the cortex. About 90% of the glomeruli are these cortical glomeruli located in the outer two-thirds of the cortex. These receive about 90% of the renal blood flow, and as blood leaves the glomerulus, some has been filtered, the blood then flows on through the peritubular capillaries, and these peritubular capillaries remain in cortex. They surround the proximal convoluted tubules and the distal tubules, and they pick up a large amount of reabsorbate and return it into the blood. At the same time, they're providing nutrient and oxygen and removing waste. The vascular architecture in the medulla is completely different. Here, these deep juxtamedullary glomeruli, only about 10% of the glomeruli are very deep, close to the junction with medulla. As blood flows through these, there's some filtration, and then the postglomerular blood plunges down into the medulla and forms these hairpin loops, these vasorecta. About 10% of the renal blood flow flows through the medulla, but only about 1% goes to the inner medulla through these vasorecta blood vessels. So we talked about the countercurrent flow in the descending and the ascending limb of the loop of Henle, where part of the medullary interstitial concentration gradient is generated. And here we also have countercurrent flow in the vasorecta. Here's the descending limb, and here's the ascending limb. This figure shows the principles of countercurrent exchange that go on in the medulla. Here's a vasorecta blood vessel, and this is the descending limb of the vasorecta, and the ascending limb of the vasorecta blood vessel. So in cortex, of course, the interstitium is 300, just like 300 milliosmoles per kilogram of water, just like the osmolarity of the blood, which arrives at the beginning of the vasorecta. The blood then flows down the descending vasorecta. Remember, this is a capillary, a leaky blood vessel. There's no active transport going on here. Everything that happens across the wall is passive, based on gradients across the wall of the vasorecta vessel. So this isoosmotic blood suddenly arrives into an area where the interstitium is concentrated. Because the wall is leaky, water will of course leave, moving from the dilute, relatively dilute blood to the concentrated interstitium. At the same time, solute will leave the interstitium and enter the blood. So we have water leaving and solute entering as the blood flows down towards the tip of the vasorecta, towards the inner medulla. This results in an increased osmolarity of the blood as it descends down towards the inner medulla. Now, unlike the tubule, even though vasorectal blood flow is quite slow, the flow is faster than in the tubule, so there's not adequate time for equilibration of the blood osmolality within the vessel and the outside interstitium. So as the blood gets to the hairpin loop, it's still a bit dilute relative to the interstitium. Now blood flow moves up the ascending limb of the vasa recta. Again, the blood is dilute relative to the concentrated interstitium until it reaches this equilibrium point where interstitial and blood osmolalities are equal. After that, as blood continues towards cortex, we now have a reversal of the gradient across the wall, with the osmolarity of the blood being higher than the osmolarity of the interstitium. So at this point, water is taken up and solute is returned to the interstitium. As blood leaves the medulla, two things have happened. The osmolality of the blood is higher of the blood leaving compared to the blood entering, and this reflects the fact that some solute has been taken up and removed from the interstitium. This is necessary because although we need a standing gradient due to sodium and urea trapping in the interstitium, excess sodium and urea must be removed. The second thing is that the rate of blood flow leaving the vasa recta is higher than the rate entering, and that reflects the fact that excess water has been removed from the interstitium to prevent the water from diluting the interstitial concentration gradient. So remember that this is a steady state. We've got continual accumulation of sodium, chloride, and urea in the interstitium. We're delivering blood, and because of this countercurrent exchange, there's some recycling of the solute, so that not all of the solute in the interstitium is flushed out. Imagine what it would be like if the blood entered down the descending limb and then left at the tip of the papilla. A large amount of solute would be flushed out, and it would be impossible to maintain a medullary interstitial concentration gradient. When vasa recta blood flow increases significantly, there is also some flushing out of excess solute, and this leads to a reduction in the medullary interstitial concentration gradient. So we've described how the gradient is established, and now how it's maintained while at the same time removing excess solute and water and waste by the blood. Now let's go back and look at a tubule with an established medullary interstitial concentration gradient and see how a concentrated urine develops when ADH is present. During passage along the proximal tubule, there's isoosmotic reabsorption of sodium and water, so that the osmolality of the fluid arriving at the beginning of the descending limb of the loop of Henle is 300 milliosms per kilogram of water, the same as the cortical interstitium. Fluid then enters the descending limb of the loop of Henle, which is very leaky to water, and that's the only event which occurs across this segment of the tubule. Water leaves. As the water leaves, the solute that's trapped behind gets more concentrated. And because this is such a slow flowing system, we see near equilibration by the tip of the loop of Henle, at the hairpin bend, with the interstitium. This solution now contains a very high concentration of sodium. Sodium flows up through the thin ascending limb of the loop of Henle, and there's a little bit of outward diffusion passively of sodium down the concentration gradient from the very high intratubular sodium to the relatively dilute interstitial sodium. The tubule fluid arriving at the beginning of the thick ascending limb is extremely concentrated with regard to sodium, and this stimulates the NKCC2 sodium chloride transporter in the thick ascending limb of the loop of Henle. By delivering an increased concentration or quantity of sodium, these transporters are stimulated to transport more sodium out. In fact, the thick ascending limb NKCC2 transport is so vigorous that by the time the tubule fluid gets to the top of the thick ascending limb and enters the distal tubule, it is dilute relative to the rest of the body fluids. Fluid then flows along the distal tubule. There's continual sodium reabsorption, now under the action of aldosterone, by the NCC, sodium chloride cotransporter. No water leaves, and by the time the fluid gets to the beginning of the collecting duct, it's even more dilute. Always, under every circumstance, whatever the final urine osmolality, you will always see dilute fluid entering the beginning of the collecting duct. Tubule fluid now begins to flow down the collecting duct, first down the cortical collecting duct, all the way down the collecting duct. Sodium is reabsorbed now by the epithelial sodium channel, the ENaC, also under the control of aldosterone. In this segment, the control of sodium absorption is completely dissociated from the control of water absorption. In this example, antidiuretic hormone is present, which means that the whole collecting duct is very permeable to water. Water leaves, some sodium leaves, but there's very little sodium left at this point. And by the time the fluid leaves the inner medulla and forms urine, the urine osmolality has risen as high as 1200 milliosms per kilogram of water. So that's 4 times the concentration of the fluid in the proximal tubule or the interstitium or the general body fluids. Now let's look at what happens when ADH is absent. The first thing to notice is that while the cortical interstitium is still the same, 300 milliosms per kilogram, the medullary interstitium is concentrated, but the concentration gradient is less than when ADH is present, because there's no longer a urea reabsorption and recycling mechanism. That is absent. So the only contributor to this medullary interstitial concentration gradient is absorption of sodium chloride from the thick ascending limb. Again, the tubular fluid is about 300 milliosmoles per kilogram of water as it enters the descending limb of the loop of Henle. Water will leave down the osmotic concentration gradient because the descending limb is always permeable to water. That means that the tubular fluid will get more concentrated, but it can only rise to the concentration in the adjacent interstitium. So in this example, it will increase to 600 milliosmoles per kilogram of water. We've now got a high concentration sodium solution being delivered into the ascending limb. There's a little bit of passive sodium transport into the interstitium, passive sodium reabsorption, but the majority of sodium reabsorption occurs actively in the thick ascending limb, again, stimulated by the arrival of a concentrated sodium solution. The transport of sodium chloride out of the thick ascending limb is again so rapid and vigorous that the intratubular osmolality has fallen to below proximal tubule or cortical interstitial or body fluid osmolality. There's more sodium reabsorption from the distal tubule, no water transport, so again we end up with a dilute urine arriving at the beginning of the collecting duct. There's continued sodium reabsorption by the ENEC. Again, the sodium transport in the distal and the collecting duct are regulated by aldosterone, and there's no ADH present. This wall of the collecting duct is impermeable to water. In that situation, there's continued sodium removal, but all the water remains in the tubule, which means that the concentration of the intertubular fluid falls, and it can get as low as 30 to 50 milliosmoles per kilogram of water in a maximal water diuresis. We'll talk a little bit about how antidiuretic hormone elicits its hydroosmotic effects on the collecting duct. It works on the principal cells, and antidiuretic hormone has two sets of receptors. It has the V1 receptor, which are in the blood vessels, and activation of those V1 receptors causes vasoconstriction. We talked about that in response to severe hemorrhage. But these receptors, the V2 receptors, mediate the physiologic effect of antidiuretic hormone. So ADH combines with the V2 receptor on the peritubular side of the principal cell. This is a G-coupled receptor which stimulates adenyl cyclase to form cyclic AMP. Cyclic AMP stimulates protein kinase A, and the first thing that protein kinase A does is stimulate translocation of these vesicles, which contain multiple preformed aquaporins, and they're sitting in the cytoplasm of the principal cell. And it causes these vesicles to translocate to the luminal membrane and release the aquaporins, inserting them into the luminal membrane as open water channels. Additional phosphorylation events triggered by protein kinase A also result in synthesis of new aquaporins. When antidiuretic hormone levels are reduced, we see endocytosis of these aquaporins, which were sitting in the membrane. These move back into vesicles where they're stored in the cytoplasm and later recycled when stimulated with additional ADH, or in some cases, they are degraded. So in this lecture, we discussed how the combination of a very slow vasorectal blood flow and, importantly, the fact that the vasorectal vessels have a countercurrent exchange between the descending and ascending limbs, how this maintains the medullary interstitial concentration gradient, while at the same time delivering oxygen and nutrient and picking up waste from medulla. We've seen how when ADH is present and there's a high medullary interstitial concentration gradient, the collecting duct reabsorbs a large amount of water and produces a very concentrated urine. When ADH is absent, there's little water reabsorption from the collecting duct, but continued sodium reabsorption, which results in a high volume, very dilute urine. The hydroosmotic action of ADH is mediated by activation of the V2 receptors on the principal cells, stimulating cyclic AMP and leading to a rapid insertion of preformed aquaporins into the luminal membrane, as well as a slower synthesis of new aquaporins.